Namaskar, I am Vandana. Me with Bhagath Krishnan is presenting today on the topic Matrix Metalloproteinases, master regulators of tissue morphogenesis. The matrix metalloproteinases are a class of zinc proteases that aids in the breakdown of most of the ECM constituents. Additionally, MMP plays a part in processing elements that affect inflammation, cell development, and proliferation. The zymogenic form of secreted MMPs is maintained by a mechanism known as cysteine switch, which is controlled by an invariant cysteine residue that serves as a structural support for the enzyme's latency by ligating to the zinc ions in the active region. The experiment with Drosophila melanogaster population from the Himalayas exposed to the summertime conditions and their phenotypic and genotypic response tracked over 90 days, we observed allelic shifts in response to thermal changes and reduced capacity to withstand rising temperatures in high altitude insects. Types of MMPs. 28 MMPs have been discovered among various organisms, and human MMP family consists of 22 members. There are mainly 6 groups: collagenases, gelatinases, stromylysins, metrolysins, membrane-type MMPs, and other non-classified MMPs. Collagenases cleaves growth factor, various matrix proteins, and non-matrix proteins. Gelatinases, it's expressed in various cells like fibroblasts, endothelial cells, etc., and is crucial for ECM modeling. Stromylysins, they are secreted on extracellular matrices expressed by fibroblasts and epithelial cells and are involved in mammary gland development, wound healing, and immune response. Metrolysins, both members of this family, MMP7 and 26, have implications in the progression of cancer. Model organisms study MMPs. Drosophila melanogaster, DM1 MMPs helps in the development of the larval tracheae and reversal of the pupal head. DM2 MMPs is involved in the development of photoreceptors, cell rearrangement, etc. In sea urchins, MMPs regulate specific morphogenetic changes associated with early development. In zebrafish, high levels of MMP25 were found during the first 48 hours of the embryonic development. Researchers are still looking to identify and study MMPs from chicken genome. Although it is unknown how many MMPs are present, the few that have been currently identified include the homologs of human MMP 14, 15, and 60.
Non-coding RNA associated with MMPs. The non-translated RNA longer than 200 nucleotides in length are defined as long non-coding RNAs. The functions of long non-coding RNAs can be important and can affect the gene expression in different conditions like neural disorders, cancer, and even immune response. The figure shows that the gene mania interaction map of MMP in Drosophila. MMP1 interacts with protein coding gene like SHG, chitin synthase, tissue inhibitors, and metalloproteinases and MMPs too. Next, the figure shows about the cytoscope was used to display the cyto-Huber rank of MMP interactions. Like the intensity of the color indicate the rank. Red color indicate the first rank and yellow is with the least ranking. MMP in disease and development. Numerous diseases such as cancer, neurological disorders, lung disorders, and arthritis have been linked to MMPs. In MMP mutant embryo, cardiomyosin do not enclose near the whole lumen. They even though align in the midline. Although MMP are not necessary for the normal growth of the Drosophila head. Conclusion. MMP are the dynamic enzyme that activates the remodeling, shaping, and development of the tissue. MMP exert precise control on cell behavior, migration, and differentiation, and it also breaks down the elements in the extracellular matrix. MMP plays a part in pathological illness such as arthritis, cardiovascular disease, and cancer metabolites. MMP disrupt the extracellular matrix and cause uncontrolled tissue remodeling.